So today's topic is acetate pathway, and coming to the acetate pathway, it occupies the central position in relation to general metabolism, and in this the starting material is acetyl coenzyme A. So acetyl coenzyme A, it acts as a precursor for the acetate pathway. So this pathway is known as acetate pathway. Okay, so this acetyl coenzyme A, it leads to the formation of straight chain compounds as well as the aromatic compounds. Right. Coming to this acetate pathway, so here we can see acetate pathway, it occurs in mainly two routes, or we have two types of acetate pathways, that is acetate mevalonate pathway and acetate malonate pathway. So acetate mevalonate pathway, it is also known as isoprenoid pathway or HMG-CoA reductase pathway or mevalonate pathway. Acetate mevalonate pathway, it is also known as isoprenoid pathway because it leads to the formation or biosynthesis, it involves in the biosynthesis of isoprenoids. So this is also known as isoprenoid pathway. And as the mevalonic acid is produced from the acetyl CoA, so acetyl CoA, it forms the mevalonic acid, and from the mevalonic acid we get this isoprenoids or terpenoids. So this pathway is also known as mevalonate pathway, isoprenoid pathway, and there is involvement of HMG-CoA reductase. HMG-CoA it undergoes reduction and forms mevalonic acid. So this is also known as HMG-CoA reductase pathway. So all these are the names of acetate mevalonate pathway. So acetate pathway, we have two pathways: acetate mevalonate pathway, or it's also known as isoprenoid pathway, or it also known as HMG-CoA reductase pathway. Okay, so this pathway, it mainly involves in the synthesis of terpenes and steroids. So second one is acetate malonate pathway. So this acetate malonate pathway, in this both the acetyl CoA and malonyl CoA, okay, so acyl carrier proteins are involved in case of this acetate malonate pathway. Okay, both the acyl ACP and malonyl ACP will be involved in the formation of fatty acids and polyketides. The main role of this acetate malonate pathway is helps in the synthesis of fatty acids and polyketides. Okay, so this is the schematic representation of the overall pathway. So from the glycolysis we will get pyruvate, and from the pyruvate we will get the acetyl CoA. So this acetyl CoA is the starting material for both the acetate mevalonate pathway and acetate malonate pathway. So this acetyl CoA, it also involves in tricarboxylic acid cycle. So here we are concerned with these two pathways, right? Acetate mevalonate pathway and acetate malonate pathway. Okay, so in case of acetate mevalonate pathway, so this gives rise to isoprenoids and also terpenes. Terpenes are the oxygenated products. Isoprenoids, it contains just carbon and hydrogen, right? So from isoprenoids it is terpenes, and on the other hand, isoprenoids, it also produces squalene, and from the squalene we get steroids. Okay, so in case of acetate malonate pathway, so in this the malonyl CoA is involved along with the acetyl CoA. Okay, so this leads to the formation of fatty acids like lipids, waxes, fats, etc. So this is about the acetate malonate pathway. So acetate malonate pathway, so the enzyme involved is acetyl coenzyme A. So it's also known as active acetate. Acetyl coenzyme A, it is also known as active acetate, and this was discovered in the year 1950, and so further supported the role of acetic acid in the biogenetic pathways. Okay, so the acetyl CoA was isolated or discovered in the year 1950, and it is also known as active acetate. So later mevalonic acid was found to be associated with the acetate. Right. It contributes, this pathway contributes 1/3 of known secondary metabolites, which involves the biosynthesis of isoprenes and terpenoids, steroids, etc. And some vitamins, vitamin K and vitamin D. Right. So here isoprene units means which contains C5 and H8 in their structure. Okay, so isoprene units, it contains 5 carbon atoms in their structure, C5H8. Okay, so terpenoids means they are the oxygenated products, oxygenated compounds, so containing these isoprene units C5H8, but along with that it contains oxygen. Okay, so isoprenes contains only carbon and hydrogen, whereas the terpenoids, it contains carbon, hydrogen and oxygen in their structure. So next point, coming to the next point, the mevalonic acid further produces isopentenyl pyrophosphate IPP and its isomer dimethyl allyl pyrophosphate. Okay, so here starting with acetyl CoA. So acetyl CoA, it forms HMG CoA reductase, and from the HMG CoA reductase it forms mevalonic acid, and from the mevalonic acid it forms mevalonic acid mevalonate 5 diphosphate or pyrophosphate, and from that we will get isopentenyl pyrophosphate. Okay, so this forms a isomer, that is dimethyl allyl pyrophosphate. So these two are important in the formation of further isoprenoids or terpenoids. So these two main intermediates, isopentenyl pyrophosphate and dimethyl allyl pyrophosphate, set the active isoprene unit. So this involves in the formation of the isoprenoid compounds. So basically, what is the role of this biosynthetic pathway? They involve in the formation of anthraquinones, naphthaquinones, terpenoids, some glycosides like cardiotonics, deoxygenin, detoxygenin, silarin, strophanthidine. All these will be synthesized from the same pathway, and some alkaloids are also synthesized, and all these are synthesized. Some vitamins are there, vitamin D, vitamin K. Okay, they are also synthesized through this pathway, acetate mevalonate pathway. Right. So here the acetyl coenzyme A is the starting material, and from which the mevalonic acid is formed. Okay, this is our intermediate. So from the mevalonic acid pyrophosphate, we get isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. These two are also intermediates. Okay, so this involve in the reaction, they involve in the formation of isoprene units. Okay, so they are the precursors of building blocks of isoprenoids. Right. So coming to the occurrence of this pathway, so this pathway occurs in the eukaryotes and some bacteria, and even you can see this pathway, acetate mevalonate pathway, in plants as well as animals. So here you can see the structure of isoprene unit. So they are also known as hemiterpenes, which contains only one isoprene. By one isoprene means C5, 5 carbon atoms in their structure. Okay, so if the compound contains 2 isoprene units, then it is called as monoterpenoids. Monoterpenoids contains 2 isoprene units. Okay, so here you can see 1, 2, 3, 4 and 5. Okay, so 1, 2, 3, 4 and 5. Okay, so this is one isoprene unit and this is another isoprene unit. Monoterpenoids are made up of 2 isoprene units. And next is sesquiterpenoids. Sesquiterpenes, it contains 3 isoprene units. So here we have a cleavage, right? So here, so this indicates one isoprene unit, and this one more isoprene unit, and this is another isoprene unit. Okay, so the sesquiterpenes, it contains 3 isoprene units. Example is termarone and farnesol. The next is diterpenoids. Diterpenoids means it contains 4 isoprene units. So example is geranyl, geraniol and phytol. Okay, so here you can see this is one, second isoprene unit, third isoprene unit, and the fourth isoprene unit. So here also one, and this is second one, this is the third one, and this is the fourth one. Okay, so up to here it is one, and up to here it is one. This is the second one. This is the third isoprene unit, and this is the fourth isoprene unit. In the same way, this is an isoprene unit, and this is the second one, and till here this is the third one. Okay, so here this is the first one, and this is the second isoprene unit. Okay, so here this is the one isoprene unit, and this is one more isoprene unit. Okay, so now 1 and 2 monoterpenoids, 1 and 2. Okay, it contains 2 isoprene units. Sesquiterpenoids, it contains 3 isoprene units, 1, 2 and 3. Right? Diterpenoids contains 4 isoprene units, 1, 2, 3 and 4. Okay, so this is about the isoprenes. So this diagram shows about the compounds formed through or via this biosynthetic pathway, that is acetate mevalonate pathway. So from acetyl coenzyme A, so mevalonic acid is formed, and this undergoes reactions and gives dimethyl allyl pyrophosphate. Dimethyl allyl pyrophosphate and isopentenyl pyrophosphate both are hemiterpenes. It contains C5 carbon atoms. Both are isomers, right? So here dimethyl allyl pyrophosphate, it is a hemiterpenoid which contains C5 carbon atoms. So this dimethyl allyl pyrophosphate in presence of one more isopentenyl pyrophosphate, that is IPP, it gives geranyl pyrophosphate. So C5 plus 5 monoterpenoid, it contains 2 isoprene units, and this further in presence of isopentenyl pyrophosphate, it gives farnesyl pyrophosphate which contains 15 carbon atoms, and this on further it gives geranyl pyrophosphate which contains 20 carbon atoms, and this farnesyl pyrophosphate on the other hand it gives squalene which is a triterpenoid, that is it contains C30 carbon atoms. So this gives you the steroids, and from geranyl pyrophosphate we get polyisoprenes. On further addition of isopentenyl pyrophosphate we get polyisoprenes, and from this geranyl pyrophosphate we get carotenoids also. In presence of two molecules of geranyl pyrophosphate we get carotenoids which contains 40 carbon atoms. So this is the isoprenoid pathway or acetate mevalonate pathway. So here we can see this is acetyl CoA. So here what happens is two molecules of acetyl CoA involved in the reaction and gives you acetoacetyl CoA. Okay, so here two compounds, two acetyl CoA compounds are involved in the reaction to acetyl-CoA gives you acetoacetyl-CoA. Okay, so this in presence of one more acetyl-CoA and in presence of HMG-CoA synthase enzyme, it forms HMG-CoA. That is nothing but beta hydroxy beta methyl glutaryl CoA. Okay, so acetoacetyl CoA in presence of one more acetyl CoA and HMG CoA synthase enzyme, it forms HMG CoA. That means hydroxy methyl glutaryl, HMG, hydroxy methyl glutaryl CoA. Okay, so this undergoes reduction in presence of HMG CoA reductase and gives mevalonic acid. Okay, so this is mevalonic acid. So this mevalonic acid in presence of ATP, so the ATP, it gives a phosphate group to the mevalonic acid and will be converted into ADP. Okay, adenosine triphosphate is converted into ADP, right, and forms mevalonate 5-phosphate because the ATP, it is giving one phosphate group to the mevalonic acid. Okay, so this reaction is catalyzed by an enzyme called mevalonate kinase. Okay, next reaction, same in presence of ATP. So the adenosine triphosphate again gives one more phosphate group and can get converted into ADP, adenosine diphosphate, and this converts the mevalonate 5-phosphate to mevalonate 5-diphosphate, or it is also known as mevalonate pyrophosphate. So this reaction is catalyzed by mevalonate phosphate kinase, right? So acetoacetyl CoA in presence of acetyl CoA, it is forming HMG CoA in presence of a HMG CoA synthase enzyme, and this undergoes reduction and forms mevalonic acid in presence of HMG CoA reductase enzyme, right? So next step is phosphorylation in presence of ATP. So the ATP, it provides one phosphate group to the mevalonic acid and get converted into ADP, and this reaction is catalyzed by mevalonate kinase. So the mevalonic acid, it takes one phosphate group and get converted to mevalonate 5-phosphate. So next step, again phosphorylation. So here in this step also, in presence of ATP, adenosine triphosphate, so it gives one phosphate group to the mevalonate 5-phosphate and get converted to ADP and forms mevalonate 5-diphosphate or mevalonate pyrophosphate. So this reaction is catalyzed by an enzyme, that is mevalonate phosphate kinase. So in the next step, the mevalonate 5-diphosphate or mevalonate pyrophosphate undergoes decarboxylation, removal of CO2 or carbon dioxide from the mevalonate pyrophosphate. Okay, so this reaction is catalyzed by mevalonate diphosphate carboxylase. So mevalonate pyrophosphate, upon decarboxylation, it forms isopentenyl diphosphate. Okay, so this isopentenyl diphosphate or isopentenyl pyrophosphate. Okay, isopentenyl pyrophosphate, this in presence of isopentenyl pyrophosphate dimethyl allyl pyrophosphate isomerase enzyme, it get isomerized and forms dimethyl allyl diphosphate or pyrophosphate, dimethyl allyl pyrophosphate. Okay, so these two are the isomers. Okay, so here in presence of an isomerase enzyme, isopentenyl pyrophosphate is converted to dimethyl allyl pyrophosphate. Okay, so here with the dephosphorylation, that is removal of phosphate molecules, it gives hemiterpenes. Okay, so because directly it contains C5 carbon atom, so it is a isoprene, right? So this here we have pyrophosphate, 2 phosphate groups are there. So if you go for dephosphorylation, So we'll get isoprene units. This is called hemiterpene. The compounds with C5 which contains only one isoprene unit are hemiterpenes. Okay, so next in dimethyl allyl diphosphate. So this in presence of one more isopentenyl pyrophosphate. So it forms geranyl pyrophosphate. Here the dimethyl allyl pyrophosphate, it contains C5 carbon atoms, and isopentenyl pyrophosphate, it contains one more C5 carbon atoms. Both are isomers, so they will have the similar molecular formula. So here also it contains C5 carbon atoms, and isopentenyl pyrophosphate, it also contains C5 carbon atoms, right? So 5 plus 5. The product formed is geranyl pyrophosphate. It contains C10 carbon atoms because here it contains 5 carbon atoms and here it contains 5 carbon atoms. So the product formed is geranyl pyrophosphate, that is monoterpene. It contains C10. So two isoprene units are combined to form a geranyl. Okay, so geranyl, it is a monoterpenoid as it contains two isoprene units. This reaction is catalyzed by geranyl pyrophosphate synthase or dimethyl allyl transferase. Okay, this terpenes you will get after the dephosphorylation. You have to remove the phosphate group, then you will get this monoterpenes. Next, geranyl pyrophosphate in presence of one more isopentenyl pyrophosphate. Okay, so here now the molecular formula is it contains C10 carbon atoms, and in case of isopentenyl pyrophosphate, it is a monoterpene, so it contains C10, and it is a hemiterpene, so it contains C5. Okay, 10 plus 5. The product formed is farnesyl, which contains C15 carbon atoms. Okay, so here we are having two isoprene units, and here we are having three isoprene units. All the three combined to form a farnesyl pyrophosphate. So the product, it has total 3 isoprene units. So here it is a sesquiterpene. Sesquiterpene means which contains 3 isoprene units in their structure. Right? So example is capsidol. Okay. So this reaction is catalyzed by farnesyl pyrophosphate synthase or geranyl transferase. So this farnesyl pyrophosphate, now it contains C15 carbon atoms. So this in presence of one more isoprene unit. So here we have three isoprene units and we have one isoprene unit, isopentenyl pyrophosphate. So 15 plus 5, 20. So the product formed should contain C20 carbon atoms. Okay, let us see. So the farnesyl pyrophosphate in presence of isopentenyl pyrophosphate, it forms geranyl pyrophosphate. Okay, so here you can see it contains C20 carbon atoms, so it is a diterpenoid. Diterpenoid means it contains 4 isoprene units. Here we can check, right? The farnesyl pyrophosphate, it contains 3 isoprene units, and here one more isopentenyl pyrophosphate contains 1 isoprene units. So total 4 isoprene units are there. So if you go for dephosphorylation, you will get the diterpenoids. Example is abetadine. And next, so apart from this, the farnesyl, on the other hand, if you add one more farnesyl group, okay, so if you combine this farnesyl pyrophosphate with one more farnesyl pyrophosphate, farnesyl pyrophosphate means C15, right? So here we are taking one more farnesyl pyrophosphate molecule, right? C15. C15 plus C15, this C30. So the product formed is triterpenes, which contains C30 carbon atoms. Example is brasinolide, right? So in the same way, we have geranyl pyrophosphate. Here you can see geranyl contains C10 carbons, right? So geranyl, two geranyls, 10, 10. Okay, so geranyl pyrophosphate, it contains 20 carbon atoms. Now we are taking one more geranyl pyrophosphate. That means here it contains 20 carbon atoms and here it contains 20 carbon atoms. So the product formed will contain 40 carbon atoms. So the geranyl pyrophosphate, in presence of geranyl pyrophosphate, it forms tetraterpenes, that is it contains 40 carbon atoms. Example for this is beta carotene. So next, acetate mevalonate pathway. It mainly contains it mainly formed by the involvement of isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. Okay, so here isopentenyl pyrophosphate and dimethyl allyl pyrophosphate both are formed from the mevalonic acid. So these two help in the formation of the isoprene units, the basic structure of the terpenoids or isoprenoids. Okay, so here you can see they also produces vast array of monoterpenoids, sesquiterpenoids, diterpenoids, carotenoids, polyphenols, and also the compounds like glycosides, and some glycosides and some alkaloids are formed via this acetate mevalonate pathway. Okay, so this point we already discussed. So acetate mevalonate pathway, or it is also known as mevalonate pathway, or it is also known as isoprenoid pathway, as it is producing isoprenoids, and it is also known as HMG-CoA reductase pathway because there is a formation of HMG-CoA, and this undergoes reduction and gives mevalonic acid. So this pathway is also known as HMG-CoA reductase pathway. Okay, so this is an essential pathway, metabolic pathway, which occurs in the eukaryotes, bacteria, and some plants and in plants and animals. Next, so this point we already discussed. IPP and DMAPP, these two involve in the formation of the isoprenoid units. Okay, and so this also involved in the formation of cholesterol, vitamin K, coenzyme Q10, and steroidal hormones, and vitamin D is also synthesized via this pathway. Okay, so the mevalonate pathway begins with acetyl CoA and ends with IPP and DMAPP, that is dimethyl allyl pyrophosphate. Okay, so it is best known as a target of statins. Okay, statins are the class of drugs which lowers the cholesterol level. Okay, so this generally the statins inhibit this HMG CoA reductase within the mevalonate pathway. Okay, so they prevent this formation of all mevalonic acid and everything. Okay, so the statins are they target this pathway, mevalonate pathway, and lowers the cholesterol levels. Next, the same points are repeated. So the mevalonic acid is discovered in the year 1956. So as we already discussed, the acetyl CoA is discovered in 1950, and the mevalonic acid it is discovered in 1956, and this mevalonic acid it is nothing but 3,5-dihydroxy 3-methyl avaleric acid. Okay, so this is the pathway, acetyl CoA and acetoacetyl CoA. Okay, so first two molecules of acetyl CoA combines to form the acetoacetyl CoA, and then This acetoacetyl CoA, it combines with one acetyl CoA and forms HMG CoA. Okay, so this reaction is catalyzed by HMG CoA synthase enzyme, and then the HMG CoA is converted to mevalonic acid in presence of HMG CoA reductase. So this mevalonic acid undergoes phosphorylation in presence of ATP and forms acetate mevalonate 5 phosphate, and again it undergoes phosphorylation and forms mevalonate pyrophosphate, okay, diphosphate at the fifth position. Okay, so mevalonate undergoes phosphorylation and forms mevalonate pyrophosphate. Right? So this is converted to isopentenyl pyrophosphate, and the isopentenyl pyrophosphate undergoes isomerization and forms dimethyl allyl pyrophosphate. So that dimethyl allyl pyrophosphate in presence of one more isopentenyl pyrophosphate, it gives farnesyl bisulfone. This is the scheme, right? So the farnesyl pyrophosphate, from the farnesyl pyrophosphate we get squalene, from the squalene we get lanosterol, and ultimately it forms cholesterol. Okay, so like steroidal hormones, vitamin D, and bile acids. So here you can see the some of the secondary metabolites produced or synthesized through this pathway. So we get squalene from here, right? From farnesyl we'll get squalene. Okay, so these are the products obtained from the squalene. Okay, so till here it follows the acetate mevalonate pathway. Okay, from the acetate mevalonate pathway we get squalene, and from the squalene we get cholesterol, and from cholesterol we get 2022 dihydroxy cholesterol, and from which we get pregnenolone. Okay, so we'll see the products obtained through this pathway. So from squalene we get pentacyclic triterpenoids along with the cholesterol. Okay, pentacyclic triterpenoids, it gives beta amirin, and from cholesterol diosgenin and yamogenin are synthesized, and diosgenin in turn it gives tigogenin and yamogenin, and it helps in the synthesis of sarsasapogenin. Okay, so we also have dihydroxycholesterol, which gives pregnenolone. Pregnenolone, in presence of 3 carbon units, it forms silarenin, silaglucosidin, and helibrigenin. This, on the other hand, in presence of 2 carbon atoms, it forms digitoxygenin, digoxygenin, gitoxygenin, and strophanthidine. Okay, so these are pharmaceutically important. All these are the cardiotonic drugs. Digitoxygenin and digoxygenin are found in the digitalis. Gitoxygenin also, it is found in the plant digitalis, which is a cardiotonic used in the treatment of cardiac disorders. Okay, it acts as a cardiotonic. And strophanthidine, it is obtained from strophanthus. It also has cardiotonic activity. Diogenin, it is a glycosapine glycoside, and silarenin, it also has cardiotonic effect, which is obtained from the squill. So next pathway is acetate malonate pathway. So in acetate pathway we have both the acetate mevalonate pathway and acetate malonate pathway. So next, coming to acetate malonate pathway. So acetate malonate pathway, it mainly involves acetyl CoA and malonyl CoA. Okay, so here they function. It operates functionally with the involvement of ACP, which is known as acyl carrier protein. Okay, so this acyl carrier protein yields fatty acyl thioesters of ACP. Right. So here we have both the acetyl CoA and malonyl CoA. From acetyl CoA we will get malonyl CoA, and the acetyl CoA, it will be converted to acetyl acyl carrier protein, acetate uh, acetate ACP. Okay, and malonyl CoA will be converted into malonyl ACP. So this acetate acetyl ACP and malonyl ACP, they involve in the further reactions, right? So these acyl thioesters forms the important intermediates in the fatty acid synthesis. Okay, so these acyl carrier proteins, they yields in the fatty acid acyl thioesters. Okay, these acyl thioesters of that fatty acids forms intermediate fatty acids. So here we can see so this is the acetic acid. So this is converted to acetyl CoA. So now this same acetyl CoA in presence of carbon dioxide and adenosine triphosphate, and it forms malonyl CoA. Okay, this reaction is catalyzed in presence of biotin and manganese. Okay, so the so-called formed malonyl CoA and acetyl CoA will be involving in the reaction. Right, here you can see the acetyl CoA, and this is malonyl CoA. So this will be converted to acetyl ACP, acyl carrier protein, and malonyl CoA is converted to malonyl ACP. Okay, so these two involve in the reaction and forms acetoacetyl ACP. The acetyl ACP and malonyl ACP, they forms together acetoacetyl ACP. So this undergoes further reactions and so many intermediates are formed here, and finally forms palmitoyl ACP, right? So which gives you palmitic acid. So that is, it contains 14 carbon atoms. Next, this palmitic acid, it gives stearoyl ACP, stearic acid, which contains 16 carbon atoms, and here you can see this on further dehydrogenation, a double bond is formed. Okay, initially we are getting the saturated fatty acids. So this on dehydrogenation, it gives you the unsaturated fatty acids. So in case of folic acid, here you can see it is forming a double bond, right? So this on further dehydrogenation, it gives two double bonds. And this on further dehydrogenation gives linoleic acid and linolenic acid, which has 3 double bonds, and linolenic acid upon dehydrogenation, it gives arachidonic acid, which has 4 double bonds.